Modulation of heat shock proteins on inflammatory cytokines. Heat shock proteins are highly conserved molecules and are present in all living species, including human. Heat shock proteins are categorized into several families based on their molecular weight. In most of families, there are more than one member. They are localized in different intracellular compartments, such as cytosol and mitochondria. Heat shock proteins are constitutively expressed in normal conditions, and also they can be induced by different natural or non-natural factors such as fever, infection, as well as the vaccines. Heat shock proteins are well known as intracellular molecular chaperones or proteases. As chaperones, they take part in the assembly, stabilization, folding, and translocation of proteins. And also they can bind to the misfolded protein during the synthesis process and refold it into a native proteins which have normal functions. Today I want to talk about the function of heat shock proteins on inflammation and inflammatory cytokines. Inflammation is a beneficial host response to foreign challenger or tissue injury that leads to ultimately to restore and maintain the homeostasis. Cytokines are a group of soluble proteins, peptides, or glycoproteins. They are regulators of host response to infection, immune response, and inflammation. And they are categorized into pro-inflammatory cytokines such as TNF-alpha and anti-inflammatory cytokines such as interleukin-10 based on their functions. The extracellular heat shock protein 60 or heat shock protein 70 can bind on the receptor on the surface of immune cells and activate the immune response, including the activation of NF-kappa B signaling pathway to induce the expression of pro-inflammatory cytokines such as interleukin-12, TNF-alpha, interleukin-1 beta. After release, those cytokines can induce inflammation, including causing the fever. They can also increase the migration of immune cells such as macrophages or neutrophils into the inflammatory site. Some of them are involved in the proliferation, differentiation, and activation of some other immune cells such as natural killer cells. In contrast, the heat shock proteins produced in the cells called self-heat shock protein can inhibit the pro-inflammatory cytokines production. They activate the self-heat shock protein-specific regulatory T cells to produce the interleukin-10 to inhibit immune response, including the production of pro-inflammatory cytokines. As an anti-inflammatory cytokine, interleukin-10 can diminish the activation, maturation, survival of several immune cells such as mast cells and dendritic cells, and then reduce the production of pro-inflammatory cytokines. It can activate interleukin-10 secreting regulatory T cells to produce the more IL-10. In summary, besides maintaining of protein homeostasis in the stress condition, heat shock proteins such as heat shock protein 70 play important roles in both stimulating and inhibiting pro-inflammatory cytokines production. Did you get stress from this video? If you are interested in this topic, please send me an email, then we can discuss more about this. Thank you.